What is up everyone, welcome back to this channel. How are you all doing? Hope you are all doing fine, because today we are going to be discussing about chapter number 3. Now in chapter number 1 we discuss how our gonads develop, how our sex cells develop, and in chapter number 2 we learned how they come together and mate from day 1 to 6, and how on day 6 the embryo implants in the uterus. Now chapter number 3, this one should be a fairly easy one, because chapter number 3, the development of bilaminar embryo, think of it as an transitional chapter, because week 2 development of the embryo, there aren't that many major changes, but we need to learn this to go through chapter number 4, which is development from week 3 to week 8. So this should be a fairly easy one, this should be a short one. Hopefully I will again make it easy for you guys to understand, and let's just jump right into it. So as you can see here, this single image is all we need for this chapter. That's all, that's it. This video isn't going to be that long. This single image, that's all we need. Now we discuss in chapter number 2 how the embryo implants itself. Remember cytotrophoblast and syntheotrophoblast, remember them? Yes, the first thing we're going to be focusing on this image is the same things. Here as you can see here, I hope you can see the mouse. As you can see here, all these villi, the extra extended villi, this is the cytotrophoblast, the one we discussed in chapter number 2. If you haven't watched that video, I really would suggest you guys to watch these videos in series, chapter number 1, 2 and then chapter number 3, because I do really discuss in depth. As you can see, all these extended villi, that is the cytotrophoblast, and the one, all the white lining surrounding it, that is the syntheotrophoblast. Like discussed in chapter number 2, cytotrophoblast divides by the process of mitosis, while syntheotrophoblast does not divide by the process of meiosis or mitosis. It does not divide at all. So cytotrophoblast is the one that's gonna be dividing. All this dark orange villi, the extended villi, that is cytotrophoblast. Here it should be here. As you can see here, it is all the cytotrophoblast, while all this white edges, the layer around is the syntheotrophoblast. Now what do we see around syntheotrophoblast implantation and HCC? Like we discussed chapter number 2, implantation, syntheotrophoblast and cytotrophoblast just erode the wall of endometrium and implants the embryo on day 6. So HCG, as discussed in chapter number 2, is an early indicator of pregnancy. Now the normal amount of HCG is right here. You'll have as a reference in the report, so no need to worry about that. But what would the low level of HCG mean? It would mean either the mother is having a miscarriage, or the mother is going to have an ectopic pregnancy, which again we discuss in chapter number 2. Now higher level of HCG would mean either the mother is having multiple pregnancies, which would mean either twins or that, or the mother has an hydatidiform mole. Now back to this image. As you can see, all the cytotrophoblast and the syntheotrophoblast, but as you can see, they grow here, and you see all this, they grow here, they erode through the wall of endometrium, and then they connect through the vessels or the arteries or all the blood system of endometrium, and as they progress, as they erode through it, they leave all this, as you can see, the red lining, they are called the lacuna spaces. They are the blood vessels from the endometrial, as you can see, the endometrial blood vessels from the mother. And another thing to notice here is the cytotrophoblast after it's developed, and on the other end of the cytotrophoblast is the endometrium blood vessels. Now the embryo will have a blood system of its own. It will develop a blood system inside the cytotrophoblast. So the cytotropoblastic blood vessels will be right here, and on the other end of it would be the endometrial blood vessel, and it is only divided by a cell, few layer cells, so they can exchange blood nutrients from one system to another, from the mother's endometrial blood system to the child's embryonic blood system. They can exchange carbon dioxide, they can exchange oxygen from the mother's to the embryonic blood system. Oxygen can just pass through, and the carbon dioxide can just pass from the child, the embryo, to the mother. so they can just develop a system which can exchange particle, not all of them but only particular some particles like nutrients, glucose, stuff like that. So that's that. Now let's move inside. Extra embryonic mesoderm. Remember the embryoplastic cells, blast cells which would go on to differentiate into different things. Blast, yeah, embryoplastic cells, they here divide into two different types of cell, hypoblast and epiblast, two different by layer. Wow, that must be the name of the chapter then, huh? And the mesoderm is also divided into two different parts. Somatic mesoderm. Somatic meaning outer body, pertaining to the body. So the outer body as compared to this, the inside part, the somatic mesoderm would be the outside mesoderm, and the visceral mesoderm, which would be the inside one. So the mesoderm containing this yolk, covering this yolk sac and amniotic cavity, that mesoderm is the visceral mesoderm. Learn the difference. Visceral meaning the inside and somatic meaning pertaining to the body or the outside. So the covering layer is the somatic mesoderm while the inside is the visceral mesoderm. Now moving on to this, the embryoblast cell divides into two different types of cell, hypoblast and epiblast. Now let me enlarge it for you and rotate it. Why is it hypo and epi? Let me explain it to you. By the power of magic. See, it's turned. Just kidding, you were just editing. But as you can see here, the hypoblast and the epiblast. The analogy, hypo as in the below one, the one below, and epi as in the one above. The epi is the blue one and hypo is the yellow one. Now as you can see that they are on top of each other. The hypoblast will form the yolk sac and the epiblast will form another cells, but as you can see here they do not touch each other but on one spot, as if someone just punched them. Okay, maybe not that violent, but someone just pinched them. Let's just say someone just pinched them in one place. As you can see, the arrows, this shows the pinching, the only place where they touch, and that place, the precaudal plate, is an important one. Why? Because it is where the future mouth of a person is going to be. So now we know where the mouth is. It will really help knowing where and which cells is going to become what. So every cells around the precaudal plate, now we know that it's going to be forming all the head system and others are going to be form the rest. Now let me tell you a secret. Come here. The hypoblast and epiblast, they are two different cells, but all the cell system in a human will only be formed by epiblast. Hypoblast is useless. I mean, not actually useless, but all the different cell system, all the entire person is going to derive from epiblast. But that is for another chapter. This chapter just ends here. I told you it's gonna be a short one, but I hope you guys enjoyed it. I hope it was deep enough, because let's be honest, there wasn't really much to go through this one. But yeah, that's all you need to know from this chapter. I hope you guys enjoyed it. If you have any questions, ask them in the comments below. I hope you like it, and I will see you in the next chapter. Thanks and goodbye.